Hello, this is Dr. Dan Guerra. I come to you from Authentic Biochemistry Studios on the 11th day of April. The year is still 2021. I know it's hard to believe. I'm going to get right into this because I have a couple of other things I have to get done today, and I want to get this lecture in. So let's just go right to it, shall we? All right. Again, Authentic Biochemistry, and we're going to be talking about lipoproteins, human aging. I'm going to give you some details. If you had paid attention to my Facebook page, I discussed this whole concept of nihilism. And so I'm giving you there the logo that I've been going to use for today's lecture anyways, which is to stamp out nihilism, which is to stamp out any kind of theory that suggests that we can know something, particularly we can know the truth from a lie, because we can know the truth from a lie. And I'm all about that here at Authentic Biochemistry. So let's get started. Okay. Now, this is lipoprotein metabolism, and this is giving you a little bit more detail than what I did before, and that's what I'm here to do right now. So let's start off from dietary triacylglycerol coming into the small intestine, okay, right here. Goes to the lymphatic system, and it ends up preparing the synthesis of our first lipoprotein called the chylomicron. This is a nascent chylomicron. It has the apolipoprotein B48. It also has the apolipoprotein A. So nascent chylomicron contains those two apolipoproteins, right? The second thing that happens is that apolipoproteins E and C are transferred to the chylomicron from a Hdl fraction that's already in circulation. So one of the jobs of the HDL, high density lipoprotein in circulation, remember HDL was synthesized from the liver, whereas we're telling you the chylomicron is put together, apolipoprotein and lipid fraction, essentially from the small intestine, from dietary lipid, right? So the HDL is already in circulation. It has apolipoproteins associated with it, E, A, and C, as shown here. Now it's going to transfer to the nascent chylomicron, both C and E, right? Remember, this whole thing, this lipoprotein fraction is not covalently associating polypeptide with lipid. This is a non-covalent association, right? This is very important to understand how you coalesce various apolipoproteins into a lipoprotein fraction with the correct stoichiometry of lipids, neutral lipids like triacylglycerol, phospholipids, including glycerophospholipids and sphingophospholipid, as well as cholesterol esters, right? All of that is packaged together, and with the appropriate density of lipid fraction to protein fraction, you generate a chylomicron. Now, the chylomicrons have to have those signature apolipoproteins coming from the intestine, and those are the B48 and A, right? So now you understand it. All right, so you pick up now these multiple fractions coming from HDL. You got ApoC and ApoE coming on. Now you have this chylomicron, which is no longer nascent, right? It has B48, E, C, and A, right? So chylomicrons deliver the triacylglycerol to the periphery, other than the liver, particularly to the adipose, because the adipose is for storage depot fat, usually in the viscera, and of course to the muscle, because the muscle is going to use triacylglycerol for hydrolyzing that triacylglycerol into fatty acid, and the fatty acid being first esterified to coenzyme A as a thioester in the cytoplasm, coming from the lipid droplet in the myocyte, then transferred to carnitine to move into the mitochondria, all still in now, we're talking about the myocyte, we're talking the muscle fraction, right? Then that fatty acid can go through beta oxidation, which I'm going to show you real soon here, reducing that fatty acid to acetyl-CoA, and at the same time generating reduced NAD and FAD, making NADH and FADH2. Those two nucleotides then are going to run through the electron transport chain, be reoxidized. Ultimately, the electrons that were stored in NADH and FADH2 are going to be used along with the protons that are pumped across the intermitochondrial membrane, generating a proton motive force to drive back into that mitochondrial membrane with the help of hydride carriers and electron carriers such as the ubiquinol pool, and then the various complexes containing iron sulfur centers and then ultimately copper through all the way to cytochrome C, generating then the full reduction of molecular oxygen to water and driving those protons through the proton pumping ATPase, which is complex 5 in the mitochondrial membrane, taking ADP and PI and generating ATP. Then ATP gets then translocated out of the mitochondria via a transporter that takes in ADP and transports out ATP, thus giving ATP to the cytoplasm in the muscle cell. Right? Okay, that ATP can then be used directly for the muscle to cause contraction. Okay, all right. So now you understand what the muscle is doing with intramuscular, intramyocytic within the muscle cell itself, the myocyte itself, to generate enough ATP to drive contraction until oxygen tensions start to decrease over prolonged use. Then the muscle will switch to generating lactic acid because it will no longer be able to carry out fatty acid beta oxidation, which requires molecular oxygen. Because of the phenomenon of the decrease in oxygen tensions flowing into the vascular beds, you're going to switch from fatty acid oxidation, which is long-term strenuous exercise and skeletal muscle, to the short-term use of glycolysis to generate enough ATP to get the job done, and then running the lactic acid pathway back to the liver, or the alanine pathway back to the liver, or glutamate and aspartic acid, then generating gluconeogenesis in the liver and sending glucose back into that muscle cell, right? The Cori cycle and the alanine cycle, and then just basically amino acid transport, amino acid utilization, and gluconeogenesis in the hepatocyte, right? And then fatty acids, of course, are doing something in the hepatocyte. They're also being catabolized. Fatty acids are also being catabolized via the beta oxidation in the hepatocyte, generating again NADH and FADH2. But that ATP is going to be used to drive gluconeogenesis in the liver. Right? Not something that occurs in the skeletal muscle cell, but does occur in the liver. That's how the liver coordinates bioenergetics with the skeletal muscle. Right? Yeah, just standard biochemistry, biochemical physiology. Let's go back to this now. We're talking about lipoproteins. So the adipose tissue and muscle cannot phosphorylate glycerol, so they transfer to the serum to be picked up by the liver or the kidney. That's step number 4, because you now are utilizing these fatty acids, right, because of lipoprotein lipase. So the chylomicron is going to be delivering fatty acid to the for example, adipocyte, which is the myocyte, but it's not going to be also giving up glycerol because of this inability to do this phosphorylation pathway, right? So it can't trap glycerol. All right, when much of the triacylglycerol from the chylomicron is transferred, they become the chylomicron remnant down here, right? Okay, now you see the chylomicron remnant still has the apolipoproteins, they're no longer apo until they're freed again, but they still have B48, right? And they still have E. But notice that the ApoC and ApoA has been translocated back to the HDL fraction, which is in circulation. Okay, that's how you recharge HDL so that it can provide the nation chylomicron with those necessary 2 apolipoprotein fractions to generate the mature chylomicron to facilitate the mobilization of that dietary lipid, which is what its function is, what the chylomicron's function is. So you see how HDL has a very important role here. Basically, it's a carrier of apolipoproteins to allow for the digestion of lipid coming from dietary triacylglycerol through the small intestine, through the lymphatics, generating the chylomicron, then moving through this whole pathway I'm showing you now. Ultimately, the chylomicron remnant, this ultimately is going to end up back into the liver, right? So it transfers the ApoA and ApoC back to the HDL, which it just did. And now the chylomicron remnant attaches to a liver binding site, shown here, right, LRP. And that contains hepatic lipase and fatty acids, cholesterol, and cholesterol esters are now transferred, whatever's left over, back into the liver, right? So this does so that the chylomicron can carry out this function again. But again, the process is to get rid of all of the dietary lipid as possible from the chylomicron fraction and not let the liver be a depot organ for all of the lipid that's being brought in from the diet or all the lipid that's fractionated back into the liver after it's been mobilized to the periphery. Yeah. So chylomicron plays a very important role here, almost as significant a role as all the other lipoproteins, because it's also dealing with the external lipid source, which is from the diet. See, that's a really important issue that you don't normally get when you discuss lipoproteins, but because we are authentic biochemistry and I'm an authentic lipid biochemist, you get this particular authenticity. So, all right, let's move on. Now we're going to talk about the Vldl fraction. Now the Vldl is going to be coming from the liver, okay, not from the small intestine now. Now we're talking about from the liver. It's going to have B100. It's going to have apolipoprotein C. It's going to have apolipoprotein E. It's going to have cholesterol, right? The second thing it does, once it's made and it's synthesized, this whole system is synthesized from the liver, right? Additional apolipoproteins are transferred from the HDL, just like they were in the chylomicron. This is where you pick up ApoC and ApoE. Now you have a VLDL that has all these apolipoprotein fractions, B100, C, and E, right? All right, now that's how it looks like it started out. That's ultimately how it gets replenished, okay? Remember, this is a pathway that's constantly circulating these lipoproteins, and the lipoproteins business is to get all the lipid fraction moving from the continents of the liver throughout the periphery so that the lipid can be correctly deposited, including phospholipid and sphingolipid and cholesterol, where the liver is the major reservoir of cholesterol, either coming from the diet from de novo cholesterologenesis, right, which is a major pathway in the hepatocyte. All right. So I have this fully loaded VLDL, and the VLDL encounters a lipoprotein lipase, the same thing in all the non-hepatic tissues, right? So the fatty acids from the triacylglycerol hydrolyzed by lipoprotein lipase found in the adipose, the aorta, the heart, because the VLDL can dock there because the apolipoproteins have a receptor for this, the spleen, etc. So basically all the non-hepatic tissue. Okay? What you then end up with is an intermediate density lipoprotein, which is just another name for the VLDL remnant. So we have a special name for this. We call it the IDL, right? So further loss of triacylglycerol, you're removing the triacylglycerol, right? So what you're doing is you're increasing the density because there's more protein to lipid fraction, right? The density has to do with buoyant density. So the lower the density, the more lipid. You can see the VLDL is well charged with lipid. Right? The LDLs finally now are taken up, okay? Whoops, sorry, let's go back here. Yeah, we're back here now. The LDL now is matured and it's picked up back by B100 receptors because it still is this apolipoprotein, and that's found in the liver and in some non-hepatic tissue, such as the kidney and also to some extent in muscle cells. Okay? So now you have the LDL being reabsorbed by the liver. Now it can be regenerated to come back out as VLDL. So the LDL receptor is really important, right? LDL receptor is really important for picking up LDL fraction from circulation. Now you can see here that the LDL and including the VLDL and the IDL, these are not toxic substances in normal physiology. They are involved in the transport of lipid from the hepatocyte, from the liver, to all the other locations in the periphery. Now, that's absolutely necessary to do. We are oleaginous organisms. That means, as I told you, that we store depot fat. We do that as an energy reserve, right? So we put it on the viscera, primarily. There's other places where we can store depot fat, such as in the chest and in the buttocks. That's another major place where you find a lot of adipose in humans, right? And that's all called upon a reserve, from that reserve, when you go into a fasting state. Now, remember, the human evolved in a condition where there was a lot of feed-fast interchange, and it wouldn't be in a day cycle. It could sometimes be feed and then not eat for several days, at least not a high caloric density diet with a lot of high caloric density lipid. So triacylglycerol is a really good high dense calorie source, right? But you don't get a lot of that unless you get it from animal sources. There's not a lot, unless, of course, from oil seeds. And, of course, currently we get a lot of our lipid from oil seeds like soybean, like oilseed rape, also known as canola, some sunflower, of course, and from safflower, and from olive, right, olive oil. But remember that none of these plants were grown in crop agriculture during the evolution of this process. There was no crop agriculture. The early agronomists had not taken plants from the wild, bred them so that they had high seed yield and high quality seed, with some of them being oil seeds, so that you were able to press all that oil out of the olive or out of, say, soybean. Soybean was more of a protein agricultural commodity, actually, for most of its time in agronomy, in plant physiology. It was only more recently when the United States started growing a lot of soybean in the central part of the country, in the Midwest, where high levels of triacylglycerol, up to about 20%, really, like 25%. But soybean sells a lot of protein. And soybean used to be just a protein source, but that's another story. Because soybean also doesn't just store triacylglycerol, also stores protein, right? So it's a protein source. But that aside, you see now how this whole pathway is very abundantly important, because it's transferring all of this lipid, right, to non-hepatic tissues all the time, and it's in conjunction with working with the HDL fraction, right? So all these different LDL fractions are working with HDL to pick up apolipoproteins and to mobilize lipid throughout the body. So you should never think that low-density lipoprotein is a toxic lipoprotein. It is not. The only thing that the biomedical people, when they started publishing papers on cholesterol, was that if you have oxysterol, oxygenated or oxidized cholesterol that is associated with LDL, that can induce a chain reaction of peroxidations if it is taken up by certain tissues. And that can lead to that foam cell production that we talked about with the incorporation of macrophages, for example, in the vascular bed, right? But cholesterol is a very important lipid to be transferred to all the rest of the cells, because most of the rest of the cells do not synthesize much cholesterol de novo. And you need a lot of mole percent of cholesterol in every plasma membrane of every cell in the human. So cholesterol is an absolutely vital lipid, a prerenal lipid, that is about 50% mole percent in the plasma membrane. Less so, of course, in the other membranes, Golgi, endoplasmic reticulum, mitochondrion, peroxisome, and nuclear envelope, to name a few. But the plasma lamina is about 50%. So this is not something that is unnecessary. It's absolutely vital for those membranes to function. You need cholesterol in the membrane to function. All the receptors that we talk about when we talk about the immune response require cholesterol to all the other lipids. Remember, we talked about cholesterol ceramide ratios in previous lectures, or cholesterol sphingomyelin ratios, talking about lipid membrane rafts, the transporting of membrane receptors to the surface membrane receptors that work as, for example, the insulin receptor or the GLUT receptor for taking up glucose, right? they require a certain mole fraction of cholesterol. So this is an absolutely vital, normal physiological response. I want you to understand that. There's nothing toxic about this at all. Okay. All right. So a little bit more about LDL. You have LDL receptors, and the LDL, of course, has cholesterol esters. Now it's showing you having cholesterol linoleate. Now you know that linoleic acid and alpha-linolenic acid are the two essential fatty acids in the diet because we don't synthesize them as higher mammals. The reason we don't is we don't have the appropriate desaturase enzymes, which will put double bonds from the middle of the fatty acid molecule. Let's say it's an 18-carbon fatty acid, so it's from 9 carbon towards the 18th carbon terminus, where you'd find the methyl group, coming from the number 1 carbon, which would be the carboxylic acid on these aliphatic long-chain fatty acids, right? That our system doesn't express desaturase enzymes to pump double bonds towards the end of that molecule. We put double bonds from the middle of the fatty acid, say from the 9 position, towards the carboxy terminus. Okay. Now, when you get further out into that fatty acid and you get towards the terminus of it, 3 carbons away from the terminus is where we call an omega-3 fatty acid. 6 carbons away is an omega-6. The omega-3 fatty acids are sometimes called marine lipids, which is actually a misnomer because it's not the fish that synthesize it, because fish are animals. They actually take in phytoplankton. So the desaturases that put double bonds at that end of the molecule, the omega, the end of the fatty acid molecule, are desaturases that are expressed in plants and in some microorganisms, right? And so because of that, they're essential in the diet because we don't express those kinds of desaturases. So you have a delta 12, delta 15 desaturase to be able to produce linoleic and alpha-linolenic acid respectively. And you can get that from plants, but you can also get that from fish because the fish consume phytoplankton. And the phytoplankton are the organisms that are synthesizing sometimes very long-chain polyunsaturated fatty acids like eicosapentaenoic and docosahexaenoic acids. There's also some docosapentaenoic acid generated in marine flora. Right? So let's go back to this LDL discussion here. So you've got cholesterol. This is just showing you can have a very important LDL fraction that's going to be carrying an essential fatty acid. Now, why is that essential? Because 18 carbon fatty acid with 2 double bonds, like linoleic acid, is going to be the correct chain length and positional isomer of that polyunsaturated fatty acid, right? It's going to be a dienoic. It's going to have 2 double bonds, the linoleic acid is, so that you can elongate that fatty acid and desaturate it towards the carboxy terminus. And that's going to generate, ultimately, the fatty acids like eicosapentaenoic, docosahexaenoic. And if it's an omega-6 series, it's going to, of course, make arachidonic acid. All those fatty acids are often found in the 2 position of glycerophospholipids, and upon phospholipase A2 activation, such as stress phenomena, that fatty acid can be released and then go through either cyclooxygenase or lipoxygenase, and sometimes P450 oxygenase pathways, generating the family of eicosanoids, which function in the immune response to trigger the initial phases of an inflammatory response, thus signaling a defense phenomena that can ultimately lead, of course, to the mobilization via chemokine and cytokine metabolism in the neutrophils and in the circulating basophils, eosinophils, as well as macrophages and other antigen-presenting cells like dendritic cells, to be able to carry out the innate immune response. And if necessary, ultimately, the acquired immune response by signaling using that antigen presentation to T cells, to the T cell receptor, which will then go on to activate B cells and ultimately, perhaps because of recombination, generate immunoglobulins to fight microorganismal infiltration into tissue or into circulation in humans. You see how that all works. It's very, very important that you understand how lipoproteins play a very major role in the immune response. I just explained it to you in a minute or less. Right? Okay. So LDL particles with the ApoB attach, and ApoE attaches to the LDL receptor shown here. Endocytosis of the LDL particle and the receptor all occur. Oh gosh, sorry about that. Oops. Okay, sorry. I don't know why it bounced like that. The LDL particle fuses with the lysosome. See, shown here. All right. This is all now found. This is the LDL fraction. It's still moving with its apolipoproteins. It fuses with the lysosome. Right. The LDL receptor returns to the membrane surface. So the receptor gets kicked back to the surface so it can function again. So that's a protein, right? So it can pick up more LDL in circulation, right? But then what happens is the apolipoproteins are all digested by proteases. And the proteases generate amino acids. And also cholesterol is dropped off from the lysosomal activity. And that cholesterol then can function to make a cholesterol ester, which can be stored. This is how cholesterol can then be brought into the cell. I told you cholesterol needs to be brought into most cells because there isn't enough biogenesis of cholesterol necessary for the plasma membrane for that cell to carry out its function. So this is one of the major functions of the low-density lipoprotein, is to bring cholesterol into the cell. Right? So proteins of LDL particle hydrolyzed amino acids, free cholesterol released from the LDL particle. I just showed you that, showing you through the Golgi apparatus there. HMG-CoA reductase is involved in cholesterol biosynthesis. We know this because statins attack that enzyme. When excess cholesterol is present, it inhibits HMG-CoA reductase. So the synthesis of LDL receptors also is inhibited. So that is the expression of the LDL receptor, and the HMG-CoA reductase enzyme itself is inhibited because you have high levels of cholesterol. There's no cholesterologenesis. But this is part of the feedback regulation. When a person takes a statin drug, you corrupt this normal feedback inhibition, which means that you don't only corrupt de novo cholesterologenesis from this pathway because you're blocking HMG-CoA, therefore blocking all the prenolipid synthesis, such as what? Geranyl pyrophosphate and farnesyl pyrophosphate, which could be used covalently to modify proteins at the carboxy terminus, sometimes with methylation as well, leading to the mobilization of receptors to the membrane that function to carry out all the functions of trafficking and communication in the membrane. Right? That's what's necessary about de novo cholesterologenesis that you don't get from getting preformed cholesterol from the diet. So you need that de novo synthesis, right? So excess cholesterol in the diet can inhibit that itself, but typically it doesn't have, you don't have enough cholesterol in the diet to cause anything like that because it doesn't make enough of an overload. Now you can have high levels of cholesterol in the diet, but it's not normally enough to corrupt this process. Process gets corrupted more by high levels of triacylglycerol, which crowds out the lipoprotein fractions in circulation, thus corrupting the overall lipoprotein pathway. And then the lipoprotein that gets the bad press on all this is the LDL, because it's a major workhorse for translocating cholesterol, but also triacylglycerol to the periphery, as I just showed you, through the VLDL and the IDL, the LDL, reuptake by the hepatocyte, and back out as VLDL. Remember that? Of course you do. Okay. So cholesterol is transferred to the Golgi, esterified with the acyl-CoA acyltransferase, and stored in the cell as a cholesterol ester. Okay. Good. All right. So next. So the high-density lipoprotein removes unesterified cholesterol from cells and other lipoproteins. This is canonically what we say it does, but you just saw it does a lot of other things. It also carries out all the apolipoprotein trafficking. So it returns it to the liver for excretion in bile. So that's one thing that can happen with cholesterol. Cholesterol is broken down to bile acids, okay? And that is important actually for the regular process of the final stages of the kidney function, and once again working in the small intestine and then in the large intestine, and for evacuation of ultimately what is no longer going to be used as a foodstuff, right? Excretion. So bile acids are really important for that process. But cholesterol, once it's broken down, is broken down to these bile acids, which are not going to be reused to synthesize cholesterol. So it's not like you can take acetyl-CoA from the beta oxidation of fatty acids, and actually that carbon could be potentially reused to build fatty acids. Not the case with cholesterol. It's a dead end. Cholesterol is broken down to bile acids and it's excreted, but the bile acids are super important for the completion of the digestion process in higher mammals like humans. All right. And you also have this enzyme called cholesterol acyl transferase, right, which carries out exactly that function I'm talking about. Is it's going to be able to get cholesterol away from the cholesterol ester form, so the cholesterol could be utilized in the membrane, because it can't be utilized as an ester of a fatty acid. But you also carry with that fatty acid, which sometimes can be an essential fatty acid, such as linoleic or alpha-linolenic acid. Right? Those are two 18-carbon fatty acids with double bonding that cannot be generated de novo in any human cell. Okay. Take a look at this. HDL is synthesized. Here's your HDL story now, okay? Primarily in the liver, with a lesser amount from the intestine, okay? There is some, maybe about 5%, maybe as much as 10%. So ApoE and ApoC are synthesized in the liver and added to the HDL fraction. That's the first thing that happens, shown here. Sorry about this. Mouse isn't functioning correctly. My normal mouse isn't with me right now. So here's synthesis, ApoE. This just means transcription translation of these polypeptides, right? Now you have a nascent, also called a discoidal HDL, because of the way that it fractionates in an ultracentrifuge. It's discoid in nature. So you've got phospholipid, you've got cholesterol, then you've got LCAT, right? That's the enzyme that's going to be able to move fatty acids in and out of the cholesterol pool, right? So the nascent or discoidal HDL contains phospholipids, cholesterol, and this enzyme lecithin cholesterol acyltransferase. So lecithin is another word for phosphatidylcholine, okay, which can be the source of fatty acids, because phosphatidylcholine has a 1 position and 2 position where there are fatty acids located. So phospholipids become a generator of the mobilization of essential fatty acids and also just bulk fatty acid. Okay, so third thing that happens is receptors of the adenosine triphosphate ATP binding cassette proteins, those are called ABC proteins. This one happens to be isoform A1, right? Transports cholesterol and phospholipid to the HDL unidirectionally. Okay, so this protein carries out this function. Okay, when it says ATP binding cassette, that means it hydrolyzes ATP. So it binds to ATP, and in order to carry out this physiological function, it then removes the gamma phosphoryl from ATP. So it's an ATP binding protein. It has an ATP binding cassette. So it's an ABC protein. Okay. Very important proteins in the body for multiple things like this, carrying out functions of mobilization of such things as lipoproteins and other large molecular mass fractions in and out of cells in a very specific way, right? Okay. So what else can happen is this SRB1, receptors of the scavenger receptor protein, scavenger receptor SRB1, transfers the lipids back and forth, okay, in and out of this HDL fraction, okay? So why is it doing that? Because some of this fatty acid needs to make it into the muscle and into the adipose. Now, if too much of the fatty acid is retrieved and retained, it can build up on the arterial walls and generate an atheroma. It can also generate atherosclerotic plaques. The HDL is actually capable of doing that. But because it's doing so many other things in circulation, it's not normally involved in the pathogenesis of atherosclerosis. But it can do that even when LDL is in a much larger fraction, because the HDL is usually erstwhile involved in all this other metabolism, basically moving around apolipoproteins and retro-transporting cholesterol back to the liver for bile acid synthesis, like I showed you in the last slide, via the LDL pathway. As the HDL particle picks up phospholipid, okay, so this is saying fraction 5 here, right, phospholipid, and cholesterol esters, a polar bilayer of phospholipid is formed with a nonpolar core of cholesterol and cholesterol esters. So the phospholipid, it basically starts forming a phospholipid membrane-like structure around this particular lipoprotein fraction. This is how actually membrane biogenesis prebiotically very well started out, because you get an amphipathic molecule like a phospholipid, which will normally coalesce because of biophysical interactions with the neutral lipid, and then whatever aqueous is trapped inside that lipoprotein in conjunction with hydrogen bonding with amino acids that make a part of the apolipoprotein fraction, right? And so some of those anionic and cationic amino acids are playing a major role here for titrating that hydrogen bonding to generate enough water around those apolipoproteins to allow for the phospholipid to migrate away from the triacylglycerol fraction, thus generating this mature form of an HDL. The same thing happens in a protocell during cellular development. Okay? So the HDL spherical particle cycles in size and lipid content by transferring cholesterol esters, see here, to this so-called HDL3 fraction. Okay, so the HDL has the nascent discoidal form, then has this intermediate form, which was most abundant with lipid. And then because lipids are being transferred through this SRB1, which is a natural phenomenon, I told you all lipoproteins traffic lipids in and out of the periphery. But you're increasing the amount of protein here, right? And decreasing the amount of lipid, particularly triacylglycerol. So that's why it's a high density lipoprotein, because it's enriched in protein versus lipid. The ratio is being flipped. You understand? Okay, and remember this density has to do with its density, buoyant density and ultracentrifuge. That's where all these were fractionated when people first started using ultracentrifugation to isolate what was in circulation. People want to know what was in circulation. They found the lipoproteins, how they were discovered. Okay. All right. So to continue this process here, this process so-called 5, transporting back and forth lipids, right? And then the apolipoproteins being condensed, and so you make a higher density, right? So the HDL spherical particle cycles in size and lipid content by transferring cholesterol esters to the liver via that SRB1. This is mediated by an enzyme called cholesterol ester transport protein, CTEP, right? A specific protein which will move cholesterol esters, right, as the cholesterol as an ester. Phospholipid is transferred to the liver, and lysolecithin, that's lysophosphatidyl-choline. Now you've lost typically the fatty acid in the 2 position. Lysolipids, usually the phospholipase A2 is generating the lysolipid from a preformed phosphoglycerol lipid so that the 1 position remains solvent. Sometimes, though, there are phospholipase A, for example, will remove the fatty acid in 1 position. It's still a lysophospholipid, you see, only now it's a monoacyl where the fatty acid's either in the 2 position or the 1 position left over from either phospholipase A or phospholipase A2, respectively. Okay. The lifespan of the HDL is about 2 days. And during this time, it actively works the reverse transport of cholesterol because it's also doing that. It's also taking cholesterol back to the liver. This is the process. So it gets rid of all that triacylglycerol. It's utilizing a lot of that phospholipid. It's increasing in density. It's carrying all those apolipoproteins so that they can carry out and carry out a consortium attenuated effect with all the other lipoproteins in circulation, the VLDL, the IDL, the LDL, right, and the chylomicron. But at the same time, what's left over is cholesterol, so you ultimately get that HDL fraction being taken up by the liver, and that's what's shown here. There's your SRB1, this is your receptor, right? And so then you're taking in all this cholesterol, And some of that cholesterol can be then broken down into bile acids, and that moves through the intestine, finishing the digestion process, which I've already explained to you. Okay. Then resynthesis, re-expression of the apolipoproteins, reforming now that nascent discoidal HDL with all this, with phospholipid and free cholesterol, as well as these the LCAT enzyme, right? All of that now, and then you redo the whole pathway over again. So that's how it works. Now you understand LDL, VLDL, IDL, chylomicron, and HDL. So basically, we're finished with all the different fractions of lipoproteins. Okay. All right. So when we talk about lipids, lipoproteins, and cardiovascular disease, of interest regarding cardiovascular disease, CVD, cholesterol, oxycholesterol, actually, not cholesterol by itself, it doesn't cause any kind of inflammation. It has to be oxysterol, and we will show you that pathway eventually, how that occurs. Okay. Obviously, with molecular oxygen, the cholesterol has to react with molecular oxygen, right? You also have saturated and unsaturated fatty acids as esters to cholesterol. Trans fatty acids, which are really a something that was described in the biomedical literature for many years, but which is essentially, from my point of view as a lipid biochemist, so my dialectical analysis of the research, right, so it's not an opinion, suggests that trans fatty acids are not toxic to humans. The literature that says that was mostly done in animal models with excessive amounts of fatty acids and probably generating a lot of reactive oxygen along the way. Thus, it looked like trans fatty acids were causing pro-inflammatory responses and then potentially some kind of problem with CVD, cardiovascular disease, or even with oncogenesis. That literature is definitely in doubt. Okay, I'll just put it like that. And I will show you why. I'll show you the oxidation of trans fatty acids versus cis, which are the normal fatty acids, and they all go down the same pathway of beta oxidation, because you can move double bonds around, positionally move them around during beta oxidation, and what else you can do is you can also change the geometry. So you can go from cis to trans and trans to cis. Thus, you can break down trans fatty acids just like you can normal quotation marks, cis fatty acids. And I already told you in many previous lectures that sphingolipids have within their core, the sphingosine base, a trans palmitoleic acid, trans fatty acid. It is naturally there, okay? Always. So claiming that trans fatty acids are only produced by industry, food industry, is also completely not true. It's not that it's partially not true. It's not true. Yeah. I just want to make that clear. All right. And also you have these other apolipoproteins. Apolipoprotein E is one that has some literature about it associated with Alzheimer's disease when there's a couple of different mutations in ApoE. If that ApoE ends up in the central nervous system in the late stages of life. Okay. So it'll reappear later when we talk more about Alzheimer's disease. We put this together with the dialectical event ontology discussion of aging, the immune system, and the epigenetic retailoring of the system towards morbidity and mortality, humans ultimately leading to death. Okay. So I'm going to stop here because these slides here were made quite a while ago. They weren't made by me, and they do have some mistaken opinion about whether or not, like this first thing here, a high HDL, low LDL, healthy. That is so simplified that it's not true. So that's why I want to stop here. But I think we did okay. I think we did the basic lipoprotein lecture. We can do a lot more detail about that, as you might guess, because all those apolipoproteins have their own history and story about how they're functioning, not just in lipoprotein metabolism, but carrying out other events in the cells they're synthesized in and mobilized into and degraded within. Because the amino acids that make up those polypeptides can be used to resynthesize other proteins that are necessary, for example, cell cycle shift in certain activated cells. Which ones am I thinking about? That's right, the acquired immune system cells, such as T lymphocytes, my favorite cells. All right, so I'm going to stop here. Let me see if I can find got to get rid of that first. That meaning that image of me. There we go. All right. So I'm going to leave you with this discussion, and we're going to get back to it because there's a lot more to say. We're now into the depths of lipid metabolism. I'm going to talk to you next about fatty acid metabolism. So you brought all this triacylglycerol into these cells, right? Including cholesterol. If you brought in fatty acids, you brought in cholesterol esters, all of that lipid is going to be metabolized in those cells. Adipose is going to be mostly storage, but that's not the only thing that happens. But in the rest of the cells, a lot of other biological activity is going to occur, such as membrane biogenesis. So to understand that, you have to understand fatty acid metabolism, both anabolism and catabolism, and of course, utilization. Okay, so bioenergetics, membrane biogenesis, and signaling trafficking are going to be the three next things ordered on our lipid plate. So this is Dr. Dan Guerra. Today is the 11th of April, 2021, and this is the Authentic Biochemistry video lecture podcast. And I'm saying bye for now.